Good morning all of you. Myself Kushi from Plus One Science from GRG National Girls Senior Secondary School. Welcome to the amazing world of cellular biology where molecule wages force to maintain life. Today we will explore the rise of ATP, the energy currency of the cell. What is ATP? ATP acts as an energy carrier in the cell. It is composed of adenine, ribose and 3-phosphate group. ATP stores energy in form of ATP bond which is released when energy is needed. ATP is equals to ADP plus pi plus energy, the gradient force. In cellular respiration, ATP is produced through the process of chemiosmosis, where cell utilizes energy from the sun or food molecule to produce or generate proton gradient. The gradient has two types. First gradient type is proton gradient. Its location is in mitochondrial inner membrane and its function is to produce ATP. Second gradient is electrochemical gradient. Its location is in cell membrane and its function is to regulate ion balance. The rise of ATP. ATP is important for various cellular processes including muscle contraction or ATP replications, DNA replication. The demand of ATP is high and cells have evolved efficient mechanism to produce and regulate it. Key players: ATP synthase. This enzyme is responsible for ATP production. Mitochondria, the powerhouse of the cell, generating most of the ATP. Cellular respiration, the process of generating energy from the food molecules. ATP synthase, a step-by-step process. Proton gradient formation. Energy from the sun or food molecule is used to pump proton gradient across cell membrane or mitochondrial inner membrane to produce proton gradient. And ATP synthase activation. This proton gradient drives ATP synthase to bind ADP and pi. ATP production. ATP synthase catalyzes the phosphorylation of ADP and pi. ATP release. ATP is released into cytosol where it is used to fuel cellular processes. Regulations of ATP production. First, feedback inhibition. High ATP levels inhibits ATP synthase, reducing ATP production. Second is electrolytic control. ATP synthase is activated by low ATP levels and inhibited by high ATP levels. Or ATP in prokaryotics. ATP is produced through substrate level phosphorylation and electron transport chain. Second one is eukaryotics. ATP is produced through cellular respiration, photosynthesis and regeneration. Thank you. Have a nice day.